Assalamualaikum warahmatullahi wabarakatuh. Today, I will be presenting my Bachelor Degree Final Year Project 1. My name is Maisarah Binti Zaharin and my metric number is AN180026. My supervisor's name is Associate Professor TS Dr. Nur Shohaila Binti Muhammad Sunar. My co-supervisor's name is Dr. Najiha Binti Muhammad Apandi. So the title of my Bachelor Degree Final Year Project 1 is Heavy Metals Removal by MICROALGAE BOTRYOCOCCUS ALGINATE beads in greywater treatment. So the first thing is the project outline. There are nine things that are in the outlines. The first thing is the background of the study. The second one is the problem statement of the study. The third one is the objective of doing this study. The fourth one is the scope of study. The fifth one is the significance of study. The sixth one is the literature review, which is the chapter 2. So next is the methodology, which is the chapter 3. And the second last one is the expected result, which is the expectation that I have for this project. And the last one is the Gantt chart. So this is the background of the study. So the issues that surface in this study is the contaminant in greywater. But in this study, it is focused more on the heavy metals in greywater, which can affect aquatic life, also the human's health. So the solution for these issues is to use the greywater treatment using microalgae BOTRYOCOCCUS sp alginate beads, which these beads will be examined in the sample of greywater, and after 18 days it will be examined in using the system enhancement, which is using atomic absorption spectrometer AAS and using the inductively coupled plasma mass spectrometry, which is ICP-MS. So in this study, the expected finding is to know that the microalgae BOTRYOCOCCUS sp alginate beads is success in heavy metal removal. So the next one is to know the optimum value of the BOTRYOCOCCUS sp alginate beads in order to remove the heavy metals in greywater. Next, this is the problem statement of the study. So heavy metal in greywater produces changes in the aquatic ecosystem, which resulting in a loss of biological diversity and the multiplication and bioaccumulation of the hazardous chemicals in food chain. As we know, the conventional greywater treatment is highly expensive due to the chemical additives that are required during this treatment. Thus, this study is conducted to know if the biological treatment can replace the conventional greywater treatment, which is the using microalgae alginate beads. So microalgae alginate beads treatment in greywater is low cost and does not require any chemical additives. So next, it also provides cost-effective medium for removal of fertilizers and metals from greywater. It is also highly permeable in nature, harmless, and also safe to work with. Next, this is the objectives and scope of study. The first objective is to determine the efficiency of BOTRYOCOCCUS sp alginate beads in the reduction of main parameter in greywater. The second objective is to measure the absorption on the maximum loading capacity in greywater treatment using BOTRYOCOCCUS alginate beads treatment. So the scope of study is, the first one is the microalgae beads in mg/L will be optimized for the purpose of heavy metal removal in greywater. So the second scope of study is the experimental result will be fitted to the Langmuir and Frodelic isotherm model to study the equilibrium loading capacity of the biomass. So this is the significance of the study. So the first one is the microalgae BOTRYOCOCCUS sp alginate beads have a lot of promises in treating grey water by eliminating chemicals and organic impurities as well as heavy metals. The use of phycoremediation process in grey water treatment has its own benefits, such as the microalgae accomploid are non-pathogenic photosynthetic organisms and they do not produce any toxic substance. Thus, this study will establish more knowledge about the application of using microalgae BOTRYOCOCCUS alginate beads in grey water treatment. Next, I will be presenting about the literature review. There will be more explanation about this study. So, grey water is the water that are collected separately from sewage flow that originate from washing machines, bathtubs, showers, and sinks, but does not include wastewater from toilets, kitchen sinks, and dishwashers. This means the feces and urines are not including as the grey water. It is included in black water. So the diagram shows is the pathway of the grey water sources. So it is from shower, tap, bath, mop. It is also from the household chores, which is mopping, flushing, laundry, and gardening. So I will be explaining about the phycoremediation and its advantages. So firstly, phycoremediation process is the use of algae to treat waste of or wastewater. So the algae are found all over the world and have evolved to a wide range of environment. So there are some benefits that can be seen in this slide, which is the first one is it is cost effective, eco-friendly, and also a safe process. So the second one is it is effectively reduce nutrient loads and can reduce sludge formation to a very large extent. So the third one is the microalgae accomploid are non-pathogenic photosynthetic organism. Thus, they do not produce any toxic substance. So I will be presenting about the heavy metal removal by phycoremediation. This table is a summary table of heavy metal removal using two species of microalgae that had been done by previous researcher. So the type of microalgae is BOTRYOCOCCUS sp and Syndesmus sp, but this study will be focused more on BOTRYOCOCCUS sp. As we can see, the rate of removal efficiency of BOTRYOCOCCUS sp in domestic wastewater for the heavy metal zinc is 71.5%, for the iron is 51.2%, for the cadmium is 83.5%, and for the manganese is 97.2%. So this shows how efficient are BOTRYOCOCCUS in removing heavy metals in wastewater. So this is the BOTRYOCOCCUS sp that will be used in this study, but this one is not yet being cultivated into the alginate beads. So the BOTRYOCOCCUS sp is a lipid-rich green colonial microalgae that exists in freshwater, brackish lakes, reservoirs, and ponds. It also known for its ability to generate and store large amount of lipids, which particularly in hydrocarbons. So as a result, it provides an environmentally acceptable approach in grey water treatment as well as wastewater treatment. So for the methodology, this is the flowchart of the study. First one is the sampling of grey water, which the sample of grey water will be taken at Kampung Melayu Raya, Ledang, Johor. So the next one is the sampling of alginate microalgae. The sampling of microalgae BOTRYOCOCCUS alginate beads are provided by the environmental laboratory at the Faculty of Engineering Technology UTHM. So the next one is the phycoremediation process. So next one is the heavy metal concentration test, which will be using two equipment which are inductively coupled plasma spectrometry in bracket ICP-MS. The next one is atomic absorption spectrometer, AAS. So next, after the heavy metal concentration tests are done, it will be monitor absorption on the loading capacity of the BOTRYOCOCCUS SP alginate beads. Next, it will be fitted into the kinetic model, which are Fradolic isotherm and also Langmuir isotherm. So lastly is the results and data analysis. So the figure shows the microalgae BOTRYOCOCCUS SP alginate beads that has been cultivated and will be used in this study. This is the example of microalgae alginate beads. So next, this is the atomic absorption spectrometry equipment. This equipment will be used to measure quantities of chemicals element present in grey water samples. So the heavy metals that are being checked in this equipment is ferrum and magnesium. Next, this is the inductively coupled plasma mass spectrometry ICP-MS equipment. So this equipment will be used to determine the range of metal and non-metals in concentration. So heavy metals that are being checked in this equipment is cadmium, ferrum, zinc, and plumbum. So lastly is the expected results of this study. The first one is so that the heavy metals present in the grey water samples are expected to be reduced by using microalgae BOTRYOCOCCUS SP alginate beads. The second one is the optimum value of microalgae BOTRYOCOCCUS SP alginate beads loading capacity are to be achieved in this study. So the last one is the use of Langmuir and Fradolic isotherm model to the last one is the use of Langmuir and Fradolic isotherm model to measure the equilibrium loading capacity of the biomass is successful. So this is the Gantt chart of my project, and it is started from week 1, which is on the 21st of March 2021, and it ended on the 15th week on 13 June 2021. The first activity of the first week is the explanation and discussion on research with the supervisor, and it ended with question and answer session with panels. So this is the reference that are being used in the slides. So that is all from me. Thank you.